Howdy, this is Lab Report 2, Forms of Media. Group 2, Ana Martinez.
And Hannah Garcia, Microbiology Fall 2024.
Introduction. In this lab, we inoculated different bacteria on different media aseptically. We also cultivated bacteria based on aerotolerance. The purpose of this lab is to isolate, grow, identify, observe, and study the various bacteria based on its growth on different media. Significance and background information. The significance of growing different bacteria on different media is to determine which environmental conditions are the most favorable and which are the least favorable for growth. By testing how bacteria grows under different conditions, scientists can identify and understand bacteria based on how they adapt and grow. The materials. The media is listed and the cultures that we use are also listed. We also used unknown 102 and 202, inoculating loop and needle, butane burner canister, and paraffin. The methods. We inoculated the listed media with the listed bacteria. Start by sterilizing the loop or needle when the media is liquid or semi-liquid. Then open the container containing the bacteria, flame it if it is a test tube. Then dip the loop, needle, and then take it out. Flame it if it is a test tube before closing it. Then streak the plate with the technique shown below. If inoculating into liquid or semi-liquid, just dip the needle in media. Seal the plate with parafilm to cultivate the media. If obtaining bacteria from solid culture, flame, loop, needle twice. If it's liquid culture, flame, loop, needle once. For the method, we also tested air tolerance.
So here are our results and observations. The following slides are the results and observations of our cultivated bacteria on different media. The pictures are our results and the observations are based on what we recorded in our lab notebooks. So let's start off with the blood agar. So these are our results in order. For Streptococcus pyogenes, there was no growth. Klebsiella pneumoniae, there was some growth in grayish colonies with a cloudy appearance. Enterococcus faecalis C, there was also some growth of grayish colonies, but it had more of a clear appearance. And Staphylococcus aureus, there was a ton of growth of yellow colonies with a clear appearance. For our unknowns, 102, there was a lot of growth of grayish colonies with a cloudy appearance. And for 202, there was also growth, although it was not that much. Also of grayish colonies, but it had a clear appearance. CNA blood agar. For Escherichia coli, only one colony grew and it was yellow. Streptococcus pyogenes, a few colonies grew which were white. And for Staphylococcus aureus, there was also some growth of yellow colonies. For our unknowns, 102, a few colonies grew which were whitish, and there are also some dark gray ones. And for 202, there were grayish colonies with a somewhat cloudy appearance. McConkey agar. Escherichia coli, there was some growth which was clear in color and appearance and also caused the discoloration of media to yellow. Salmonella typhimurium, there was also some growth that was clear in color and appearance, which also caused discoloration of the media to yellow. And for Staphylococcus aureus, there was no growth. For our unknowns, 102, there was no growth, but 202 had a lot of growth of purple colonies with a clear appearance. Eosine methylene blue or EMB agar. For Escherichia coli, there was some growth with a clear appearance. Klebsiella pneumoniae, there was also some growth that was also clear in color, both color and appearance. And for Salmonella typhimurium, there was a lot of growth, which was also clear in color and appearance. For unknowns, 102 had no growth, but 202 grew grayish colonies with a clear appearance. Salmonella shigella or SS agar. For Escherichia coli, there was some growth of grayish colonies with a clear appearance, which also caused a discoloration of the agar media to yellow. Shigella flexneri, there was also some growth, but it was clear in color and appearance, and it also discolored the media to yellow. And for Salmonella typhimurium, it was the same. Some growth, which was clear in color and appearance, and also discolored the media to yellow. For unknowns, 102 had no growth, but 202 had a lot of growth of whitish colonies, which also discolored the media through alpha hemolysis. Bioesculin agar. For Staphylococcus aureus, there was no growth. Enterococcus faecalis also had no growth, but discolored the agar media in a black spot. And Streptococcus pyogenes had no growth. For unknowns, 102 also had no growth, but 202 had a lot of growth of small clear colonies in both color and appearance and discolored the entire agar media to dark red. Mannitol salt agar. For Staphylococcus aureus, there was no growth. Staphylococcus epidermis and Streptococcus pyogenes also had no growth each. Go on. For unknowns, 102 had little growth of clear colonies, but 202 also had little growth of clear colonies, but also discolored the media through beta hemolysis. Motility medium. Escherichia coli had growth and red color present at the top, and so did Klebsiella pneumonia. Go on.
This is the next one.
Okay. So for the unknowns, 102 had growth of cloudy colonies with a red color present at the top, and 202 also had growth of cloudy colonies with red color present. For arrow tolerance, we used two blood agars and one thioglycolate media. One of the blood agars, okay, one of the blood agars was anaerobic, and the three cultures that we used for arrow tolerance for all three arrow tolerance tests were Escherichia coli, Alcaligenes faecalis, and Clostridium perfringens. So here are the results for our first blood agar, which was anaerobic. Escherichia coli grew clear colonies. Alcaligenes faecalis also grew clear colonies, but Clostridium perfringens grew cloudy colonies. For our two unknowns, unknown 102 grew clear colonies, and unknown 202 had some growth of cloudy colonies. For our second blood agar, which was aerobic, Escherichia coli experienced growth of alpha hemolysis with grayish colonies. Alcaligenes faecalis had growth of whitish colonies, and Clostridium perfringens had no growth. For our two unknowns, 102 had a lot of growth of a whitish colony, and 202 had some growth of a whitish colony. 102 was alpha hemolysis, 202 was beta. For the thioglycolate media, Escherichia coli had some growth of white cloudy colonies. Alcaligenes faecalis had some growth with discoloration of media, and Clostridium perfringens had no observable growth but also discoloration of media. For two unknowns, 102 had no growth, but 202 had some growth and whitish cloudy colonies. In conclusion, some bacteria were able to grow with different characteristics like their colony, such as colony color and media discoloration, and the amount of colonies. The solid medium with the least amount of growth included bioesculin agar and mannitol salt agar. The solid media with some growth included CNA blood agar, McConkey agar, and Salmonella shigella agar. The one with the most growth included blood agar and eosin methylene blue or EMB agar. The capability of bacteria to grow on different media depends on the nutritional and environmental requirements. Thank you.